Okay, very good afternoon, all the participants and respected resource person, Dr. A.RAJANI Madam Garu, Assistant Professor, Head Department of Botany, Food and Nutrition, RBVRR Women's College, Autonomous, Hyderabad. Madam, please welcome. Due to the important work as a resource person, I am unable to start the sharp 1 o'clock. Sorry, extremely sorry. At the same time, uh, I invite you to present the remaining topic, beautiful topic, that is CHARA. Please. Okay, Madam Garu, am I audible?
Yes, sir. Yes.
Please share your screen.
Yeah. It's visible, sir. My PPT is visible.
Yes, madam.
Yeah. Thank you. Good afternoon to all. Today we will discuss about the chlorophyceae last type study. The name of the CHARA. In the chlorophyceae class, already you have studied the two type studies. One is the volvox. The second one is called the Idogonia. Anyone to tell what is the volvox thallus?
Xenobia, ma'am.
Xenobia. What is the thallus of Idogonia?
Colonial.
Idogonium thallus.
Xenobia filamentous, madam.
Filamentous unbranched. That is the
Unbranched.
most important. Filamentous and unbranched plant body is there. Why I'll ask in this one? Here, the same chlorophyceae member, 3 type studies are there. The first type study name is called the volvox. The second type study, Idogonium. The volvox thallus is different and the Idogonium thallus is the different. But all type of pigmentation and reserve food material is same. Reproduction also same. But in this one, here another important that one, the CHARA. The third type study name is called the CHARA. You can study to the both Idogonium and volvox, very minute structure are there. You can't see directly eye contact. You can observe only under electron microscope only. But you can this one, the CHARA, where the CHARA is there. This is the macroscopic structure. You can observe to the directly eye contact. In your laboratory time, you can observe the CHARA in the macroscopic structure. And the thallus also, CHARA thallus also very different for the other 2 algae. Here, the chlorophyceae members, same characteristics are there. The chlorophyceae characteristics all are the same characters. But the pigmentations and reproduction term, reserve food material, all are same. See, before that one, CHARA, here, the systematic position or classification is most important. CHARA also chlorophyceae member. This one is the order Charales. The family name is called Characeae and genus name is called CHARA. In here, total in this CHARA also we will learn about the CHARA after reading this structure of what is the systematic position, where it can occurrence is there CHARA, and what type of structures are there, and what is the cell structure, what is the reproduction, and what type of life cycle is there in that one. Before in that one, the systematic position, same class is same chlorophyceae, the order Charales. The family name is called Characeae and the genus name is called the CHARA. Okay, where is the occurrence? The first one is the occurrence. Before in that one, here in my PPT, some words are there, no? Stone word, whittle word. What is these names? Here, very important, the the wall box, edogonium, the cell wall made up of cellulose, you know, all things. But here, very most important, the CHARA cell wall to made up of calcium carbonate.
Thank you, sir. Thank you.
That's where calcium carbonates to made up of this cell wall. That's where the CHARA, you can touch the CHARA, very rough surface is there. That's where this name is called stone wort or brittle wort. Sometimes to ask the question like this only. What is the stone wort? Which one is the stone wort? Which one is the brittle wort? What is the why CHARA is the brittle wort? This is the made up of the calcium carbonate. That's where the you can touch the CHARA very rough. This is called the stone. Sir, Tirupati sir, mute చేసుకోరా? Tirupati sir, please mute yourself. Unmute yourself, sir. Tirupati sir. Yeah. That's where this is the main important. Okay, coming to the example, here examples also most important. CHARA vulgaris, CHARA gelaneca, CHARA nudea is the best example for the CHARA. Next one, where the occurrence. CHARA is a freshwater. All chlorophyceae members, you know, freshwater. You can observe the PPT, the picture of the CHARA diagram or structure. See, just like tree-like structure is there, yes or no? In volvox, edogonium, very minute and spherical shape or oval shape like this, no? Here, this tree-like structure is there. This is not tree, but tree-like structure is there, macroscopic. You know, all the, which one, the chlorophyceae member is the freshwater. Same like that, CHARA is also freshwater, green in color algae, found in, submerged in shallow water plants. Shallow water, ponds, lakes, ditches, and running water, slow running water. This is the occurrence. The next one, best example, CHARA blatica is found growing in brackish water. Okay, some brackish water or some high phosphates are there like this. Different types of waters also to found, that is the blatica. Next one, CHARA, next example, CHARA fragilis, found in hot springs. Another one is the fragilis, found into the, where the hot springs of water is there, in that water spring, the CHARA found. This CHARA is found mostly in hard fresh water, rich in organic matter. Rich in organic matter, calcium, and deficient in oxygen. What is that one? Calcium and deficient into the oxygen. Here, CHARA plants often encrusted with calcium carbonate. Encrusted with the calcium carbonate. And the cell wall to made up of the calcium carbonates. And deficiency here encrusted the calcium carbonate, and hence there are commonly called as that one name, stone wort and brittle wort. This is the important. Okay, you can by heart it this name. The CHARA, which algae is the stone wort or brittle wort? The CHARA is the stone wort, brittle wort. Why? The cell wall is made up of calcium carbonate. When you can touch the CHARA, very rough surface and brittle surface is there, and this one is a macroscopic algae. The CHARA emits disgradable onion-like odor. Manaki onion smell unta chudandi, phosphorus rich una smell. That's where this is the disagreeable onion-like odor due to presence of sulfur compounds. The sulfur compounds is a very rich. That's where the smell is same like that onion smell is there. And the next one, another important CHARA hati, grow in trailing on the soil. Chara nuda and Chara nuda, Chara heti, and Chara grovici are found on mountains. And Chara velvici, Chara lyodripes found in plains of this one. This is the example. And our India, in India, Chara is represented about 30 species are there. In the India, how many species are there? 30 species are which common in Indian species. One is the Chara jailanica, Chara beruni, Chara gracillus, Chara hetei, and Chara this is the Chara hetini. This is the example for the all Indian example for Chara. Coming to the structure of Chara or thallus of Chara. This one, the thallus of Chara is the branched, multicellular and macroscopic structure. What is the structure? See, 1 second, I'll show it. Chara. Picture is not there. 1 second. No? See, you can observe this one here. The thallus of Chara is the branched, multicellular and macroscopic structure. The thallus about 20 to 30 centimeter. How much of length is there? 20 to 30 centimeter in height, but often maybe up to 90 centimeter to 1 meter height also there in the Chara. Some species, Chara heti, are small. Maybe this heti is the small, meaning 1 to 2 to 3 centimeter long. Another, the plants in appearance resemble ఈక్విజిటమ్. మీకు తెలుసు కదా, ఈక్విజిటమ్ ప్లాంట్ లాగే కనిపిస్తుంది కానీ కాదు ఇది. ఈక్విజిటమ్ ఎన్స్ కారా ఈస్ కామన్లీ కాల్డ్ యాజ్ aquatic horsetail. వెరీ ఇంపార్టెంట్. మనకి ఈక్విజిటమ్ ని కూడా ఎలాగో హార్స్ టైల్స్ అంటామో, కారాని కూడా ఏమంటారు? aquatic horsetail. The thallus is mainly differentiated into the two important. One is the rhizoids and main axis. Two are most important. Rhizoid structure is there, and see, this is the rhizoid structure. Upper part is called the main axis. The thallus mainly differentiated into the rhizoids and main axis. What is the rhizoid? The rhizoids are white. You can observe the same class, but the tip, here the thallus is very different. You can't see the in Volvox and Edogonium thallus. No rhizoids, nothing is not there. Very simple cells are present. But here, the development of rhizoids and plant, the Chara height also more than 1 meter, 2 centimeter to 1 meter height also there. In this Chara, two branches are most important. The down part is a rhizoid, upper part is the main axis. What is the rhizoids? Rhizoids are white in color, thread-like multicellular. These are the thread-like multicellular, unicellular and branched type. You can observe the picture. Here, rhizoids also branching are there. Yes or no? Branch structure. The rhizoid arise from rhizoidal plates which are formed at the base of main axis or form peripheral cells of lower nodes. The rhizoids are characterized by presence of obligate septa. This is important. What type of septa? You can observe the Chara rhizoid, that is the white in color. Branching is there, and septa is the characteristic septa is the obligate septa is present rhizoid. And the where you can observe the tip of rhizoid possess minute soil particles which function as the statolith. That is function as the statolith. Soils are there. This rhizoid shows to the apical growth, help in attachment of the plant to any substratum. Any substratum meaning here that is called mud or sand. This is the any substratum meaning mud or sand in absorption of minerals and in vegetative multiplication of plants by forming bulbils and secondary protonema. This is the rhizoids. It's clear, rhizoids?
Yes, madam.
Yes, ma'am.
See, మీరు ఇక్కడ చూడండి ఒక్కసారి ఇది కారాది. నేను branches అప్పుడు I'll explain to the branches. This part is the rhizoid. This part is called the main axis. Okay. This is the rhizoid's main important enti ante the white color is the important. Obligate septa is very important. Branching is important to attachment of any substratum. This is the rhizoid importance. Coming to the second part is the main axis. The main axis, the CHARA is the main axis, meaning this is the main axis. The main axis is erect, long, branched, and differentiated in nodes into nodes and internodes. It's very important. The CHARA differentiated into the node and internode, where the internode consists of a single, much elongated or oblong cells are present. Single and oblong in cells. The internode cells in some species may be surrounded by one cell, thick, some species surrounded by many layered thick cortical cells also present. This is corticated by the T-shaped cells are present in the internodal cells. The CHARA species which cortical layer, some species is absent. Where the cortical cells is absent, E-corticulated species. Coming to the node, where the node part is there, consists of pair of cells are present. Node part is the this one node. This is the internode. This node of cells, where the node is there, consists of a pair of central small cell surrounded by, you can observe, surrounded by 6 to 20 peripheral cells are present. The central cells, peripheral cells arise from a single nodal initial cells. This is the main axis. One is the CHARA, two branches are there. The bottom is rhizoid. The main second one, upper part is called the main axis. Next one, here the CHARA branches. On branches of two branches are there. In this main axis can be divided into two branches. See, this is the CHARA picture. This is the external feature of the CHARA. In this CHARA, the bottom is called the rhizoids. This one is the main axis. In the main axis, here branches are there, no? Two types of branches are there. One branch name is called branches of limited growth. The second one, branches of unlimited growth. How to identify? And another main important main axis consists of nodes and internodes also. This main axis can be divided into two branches. One is the branches of limited growth. Second one, branches of unlimited growth. First, you can listen to the explain to the branches of limited growth. Here, the branches of limited growth arise in rows of 6 to 20 from peripheral cells of the node. Where the node is there, see, this is the node, this part is node, this part is node. In between this one, internode. Where the node part is there, this node. Where the nodes are there, in that nodes around 6 to 20 from peripheral cells of nodes, the main axis on branches of unlimited growth. That is called the branches of unlimited growth. These are also called bractlets. The branches of first order, primarily lateral leaves, these branches stop to grow after forming 5 to 15 nodes. How many nodes to stop in the limited growth? After 5 to 15 nodes are formed, That's why hence they are called branches of they stop the growth. That's why this is called the branches of limited growth. Here this is called the stipules. This is called the stipules or stipulote. The stipulotes and reproductive structures are formed on the nodes of branches. Very, very important. Which branch to produce the reproductive organs? Where the branches of limited growth is there, in that branch to produce the stipulotes and reproductive structures are formed on the nodes of these branches. Coming to the second one, this is the branches of unlimited growth, this part. Branches of unlimited growth arise from the axil of the branches of limited growth. Hence, these are called axillary branch. What is this name? Axillary branches. This one is called the axillary branches or long laterals. Another name also, long laterals. These are differentiated into same nodes and internodes are there. Where the node part is there, this one also nodes and internodes, and where the node parts are there, in that nodes bear a primary lateral. See, this one is called the axillary branches or unlimited growth, and this one you can observe the primary lateral to present the primary laterals, and these branches look like main axis. Their growth is also called unlimited growth like main axis. This is called the branches of unlimited growth. Coming to the stipulotes. What is the stipulote? You can observe the picture. This is the picture of the CHARA. Here, in this one, the basal node of the branches of limited growth develop a short oval pointed single cell outgrowth. This is called the stipulotes. This one is called the stipulotes. These are called stipulotes. In most of species CHARA burmanica, the number of stipulotes each node twice the number of primary laterals. Such species are called bistipulates. The species name is called bistipulates. In some species of CHARA nuda, CHARA buruni, the number of stipulotes each node equal in number of primary laterals. Such species is called unistipulate nodes. When these stipulotes are present in one row, each node, the species are called haplostiphones. With 2 rows. Each node is called diplostiphones. In case 2 are there, diplostiphones are present. This is the stipulotes. And coming to the cortex. What is the cortex? The many species of CHARA aspera, CHARA infirma have internodal cells, main axis ensheathed by cortex cell. Such species are called corticated species. That one name is called corticated species. The cortex consists of vertical elongated. This one is the vertical elongated narrow cell. Here the internode up of it lengthy by corticating filament developed from the upper node. This is the lower half of the internode. This filament to form. The best example, this is all the T shape. Here, example, CHARA corolina or this is the best example for the cortex cell. Coming to the cell structure. Here, the cell structure of CHARA, the main axis CHARA consists of mainly 2 types of cell, nodal cell and internodal cell. Yes or no? Both cells, no, both are the consist of cells. Nodal cells is there, internodal cells are there. The cell thickening, inner cell wall, outer pectins, haploid, discoid, starch granules, all are present. First, I'll explain to the internodal cells. Internodal cells are smaller in size and isodiametric, dense cytoplasm, uninucleate, few small ellipsoidal chloroplast, and center vacuole is not developed. Instead of many small vacuoles may be present. The cytoplasm can be differentiated, outer exoplasm, inner endoplasm. This is the internodal cell wall. And internodal cells are much elongated. Cytoplasm is very high presence of the cytoplasm also present. And the next one, nodal cells, smaller in size, isodiametric, dense cytoplasm, uninucleate, ellipsoidal chloroplast. These are all the present internodal and internodal cell differentiated. And coming to the here to expect the question, the cell, first one is the brittle word, stone word. What is the CHARA cell wall to made up of calcium carbonate? To expect the question. Yes or no? That is the most important. Now, in the vegetative time also, sorry, in the reproduction time also, to expect the question in the remaining part of the importance for the theory time. Coming to the reproduction. Here, vegetative reproduction in CHARA takes place by following types. One is the bulbils, amylum starch, amorphous bulbils, tubers, and secondary protonema. Many times to expect the question, this amylum starch, example of CHARA stigularia. This one is the best example. Many times to ask this question, where the vegetative reproduction star shape to fill with amylum star, and which example for CHARA? Stigularia is the best example. First one I'll explain to the bulbil. Bulbil are spherical, oval in shape, tube-like structure, which develop on the rhizoids. Where the rhizoids are there, in that rhizoids to develop CHARA aspera. Best example for the CHARA aspera on lower node of main axis. The next example, CHARA blatica, also best example. CHARA blatica, CHARA aspera, best example for the bulbils. These, when the mature of the bulbils, the attachment of main plants germinate into new CHARA thallus. Coming to the amylum starch, this is the most important. You can by heart it. Very important this one. Amylum in some species of CHARA stigulina on the lower nodes of main axis develop multicellular star-shape aggregates of the cells are present. These cells fill with the amylum starch, and hence it is called the amylum starch. The amylum starch do detachment from the plant develop into a new CHARA thallus. Next, coming to the second one, secondary metabolism or secondary protonema. Secondary protonema, these are all tabular and filamentous structure which are developed from the primary protonema, and this one is the basal cells of the rhizoidal. And next one is coming to the another one, bulbils, amylum. Next one, amorphous bulbils. Amorphous bulbils also. 1 second, ma. This is the bulbils, a group of many cells, very irregular shape which develop into the where the node part is there. In that node part, these bulbils are penetrating in the structures. The main plant dies under unfavorable condition. This one is the where the unfavorable condition. These bulbs survived, and sometimes to make CHARA plants return to the when the coming to the favorable conditions. And secondary protonema. Secondary protonema, these also tubular filamentous structure developed from the primary protonema. Basal cells, where the basal cell of rhizoid is there, this rhizoid to produce the secondary protonema, and like primary protonema from the plant. This also important from the examination point of view, secondary protonema also. Coming to the next one, sexual reproduction. ఇక్కడ మీకు ఏం చెప్పలేదు చెప్పండి. Which reproduction is absent? Which reproduction is absent?
Asexual one.
Asexual, very important. In this CHARA, asexual reproduction is absent. Only you can read out to the sexual reproduction. That too, sexual reproduction is oogamous type and sterile envelope. What is the sexual reproduction? Oogamous type and sterile envelope. Most of CHARA species, homothalys. Mikkels, what is the meaning of homothalism? That is the CHARA gylanica. Some species of the heterothalism. CHARA welwichi. CHARA welwichi is the heterothalism. Most of species is the homothalism. Both are found in the CHARA. Heterothalism and homothalism. Both are present in the CHARA species. And sex organs. Ikkada meeku adigi expected questions enti ante you can byhead it to the sex organs name. The sex organs arise on the branches of limited growth, limited or primary lateral. This is the nucleus above the nucleus and globule. Both are present. See, here sex organs enti ante the development is a two sex organs. In common sex organs meaning male sex organ. What is the male sex organ name? Antheridia, and female sex organ name is called the archegonia. This is very common, but here the names are different. What is the names are different? The male sex organ name is called globule. The female sex organ name is called nucleus. Very, very important. Many times to ask this question also. What is the question, ma? The male, what is the name of male sex organ?
Globule.
Female sex organ?
Nucleus.
Nucleus. And here, sexual reproduction is the advanced type of oogamous. The gametes are produced in antheridia and oogonia that are enveloped multicellular sheath formed of cells derived from the cells present below the sex organs. The specialist complex structure containing the sex organs. Globule is male sex organ name. Female sex organ name nucleus. Ivi rendu baga gurtu pettukovali. This is very, very most important. See in the picture, this one is the nucleus. Green color is there, no? That is the nucleus. This one is the reddish color is there, globule. Some species is dioecious, while the other are monoecious. Monoecious species are protandrous. What is the meaning protandrous? That is the male sex organ develop first. That is called the protandrous. That name is called the protandrous. Coming to the development of globule. Globule ante enti, then develop enti chudandi. The male, this picture you can observe. 1 second. This picture you can observe in this one. The male reproductive organ name is called globule. The globule yellow in color. It consists of different parts. The first part name is called pedicel. Ekkadundi pedicel? This one you can observe the picture clearly. Yes or no?
Yes, dark like that.
Yeah, this one. Where my arrow mark is there? This one. Pedicel. The first part name is pedicel. The globule is attached to the plant by a large cell is called pedicel. Pedicel cavity of the globule and joined with the primary capitulum. Ekkadundi primary? Joined to this one, primary capitulum. This one is called the primary capitulum. And shield cells. Second one, this is the pedicel is completed. The second part is called shield cells. Even you can byhead it these cells all names. Shield cells. Each globule is in the form of ball-like structure. This one is the form of ball-like structure. This one, ball-like structure whose wall consists of cell is called shield cells. The outer wall of each shield cells as a reddish color appear, radial outgrowth, and therefore this shield cell therefore appears is the multicellular structure. Now this each and every globule in the form of ball-like structure, the wall consists of a large plate-like Large plate-like 8 cells are called. See, shield cells number, how many number is? 8 cell. You can count it. 1, 2, 3, 4, 5, 6, 7, 8. Yes or no? 8 cells are there?
Yes, madam.
Yeah. How many shield cells are there? Cells? 8 cell. 8 plate-like 8 cells. Very important. Shield cells size also most important for the examination time. And next one is the shield cells. Outer wall of the shield cells had radial outgrowth, therefore appear multicellular structure. మనకు కనిపించడానికి ఏంటంటే multicellular. Coming to the third one, this one, manubrial cells. ఇక్కడ ఉన్నాయి చూడండి. 1, 2, 3, 4. These are 4 is called, what is the these 4? Manubrial. 1, this is the 1, this is 2, this is 3, this one is 4. What is this one? Manubrium. On the inner side of shield cells. Inner side shield cells ante where the shield cells is there? In this one. This is the outer layer of the cell is called shield cell. Inner side of the shield cell, on the inner side of shield cell, in the central is attached to elongated cell. This is called the elongated cell as called manubrium cells. This is called the primary capitulum. దీన్ని ఏమంటాం మనము manubrial cells అంటారు. The next one is the primary capitulum. Shield cell is first one, pedicell is completed. Second is the shield cell. Third one is the manubrium. Fourth one is the this one, capitulum. In between manubrium lo capitular cells ఉంటాయి. Capitulum, primary capitulum. The inner end of manubrium cells are united to form 8 isometric cells are called, that is called primary capitulum cells. ఈ capitulum cells కూడా ఇక్కడ in middle part, you can observe in middle part 8 primary, ఇవి కూడా ఎన్ని ఉంటాయి? How many are capillary cells are there? Same 8 shield cell is there, 8 capillary primary capitular cells also present. After that one, next one is the secondary capitulum. The secondary, each primary capitulum has 1 or 2 small cells towards the cavity of globule is called secondary capitular. This one is the secondary capitulum cells. And next one, these are in between the manubrium, you can observe antheridial filament. Next one is the antheridium. And attached to the primary or secondary capitulum are several branched uniserrate filaments are present. That filament name is called antheridium. It contents are metamorphosed into a single anthrozoid. This one to the single anthrozoid. And anthrozoid, each and every anthrozoid elongated, same what coiled-like structure. It bears 2 flagella is present. And the liberation of antheridia, when the mature of antheridia, the shield cells separated from each other, exposing the antheridial filament. Here, the anthrozoids are escaped through a pore. The liberation of anthrozoid maybe takes place in the morning, and they may be swim until the evening. This is the escape for the antheridias. This is a total. What is the antheridium? What is this one? Globule. Okay. In the globule structure, how many parts are there? First part named pedicell. Second part named shield cell. Third one is the manubrium. Fourth one is the primary capitulum. Fifth one is the secondary capitulum. Sixth one is the antheridial filament. Seventh one is the anthrozoid. After development anthrozoid, the liberation of antheridia in the morning and till ending. It takes place in the morning and they may be swim until the evening. This one is the total, what is the global structure and how to development of the globule. See here, the development of globule, first one globule start from the adaxial peripheral cells, where the lower nodal of branch limited growth. This is the periclinical divisions are there. Outer side, inner cell, 2 outer globule initial, and inner cells undergoes to the periclinical division to form to the two cell. One is the middle cell and the second one is the base. Initial divide, the first divisions are the transfer division to form to the two cell. One is the basal pedicel, another one is the terminal antheridial mother cell. The pedicel, where the pedicel is there, that pedicel does not, the divisions are not there present in the pedicel. Only mother cell is the divisions are there. Antheridial mother cell, this one is the another divisions are present in this one. These mother cell become a spherical and divided through two vertical division at a right angle to form to the 8 cells. That is to form a quadrate. All these cells are transfer division to form to the octagon, 8 cell structure. These 8 cells now divide periclinically to form outer and inner cells. These layers to periclinical, 3 layers of cells are present. Overall, the antheridium consists of 24 cells arranged in 8 diagonal series of 3 cells each. This one is the development of globule. Coming to the nucleus, the female fertilization, female reproductive organ name is called nucleus. It consists of same pedicel is there. This is the pedicel and oogonia, tube cells. This one is the tube cells and this one is the corona. Okay. Pedicel and where this is the pedicel. It is present the base of the nucleus, which are present central and stalk cell. And second one is the oogonium. Upper to the stalk cells, this one upper part is the stalk cells. This is the oogonium. Upper part of the stalk cell is very much elongated structure called oogonium. This oogonium, it consists of the content are transformed into a single large uninucleated. Only one nucleate is there. See, single uninucleate egg is there. Another part is called this one. This is the tube cells. The oogonium is covered by 5 elongated spirally twisted cells are present. That cell's name is called tube cells. Upper part corona, coronavirus, same like that. Here also top of the oogonia, crown like small 5 cells are present. That cell name is called corona. Here 4 important characters are there in nucleus. One is the pedicel, oogonium, and tube cells and corona cells. Same the development nucleus initial division twice transversely produced 3 cells. Lowermost cell elongated function as the pedicel. 3 cell ga divide avutundi. Lowermost is the function as the pedicel of the nucleus. The middle one, the middle cells divide vertical to form 5 sheath shells. 5 sheath initial surrounding the central one. The terminal cell, oogonial mother cell. Where the terminal is there, oogonial mother cell. This mother cell divide transversely, lower small stalk cell, upper oogonium. The oogonium is enlarged in size and metamorphosed in single egg is present. The sheath of the cell, the sheath initial elongated and divided in the transfer division. 2 types of 5 cells are present. The function of corona's the total 2 types of cells are formed. The cells of the upper tier cells, upper 5 tier cells aim to modify into function as the corona cell, and these are formed the corona of the nucleus. The lower cells is there, no, in the lower part cell. This lower tier of cells is the tube cell to coming to the modify into the tube cell. The tube cell elongated and twice spirally clockwise directions around the oogonia. See, it spiral ga unnai chudandi. That is a tube cells. The egg become filled with the starch and oils. Its nucleus migrate towards the lower side. The reproductive spot develop at the top of it. This is the development of the nucleus. And fertilization, when the nucleus is mature, the spiral, you can observe this one. When the nucleus is mature, the spirally twisted tube cell separated from each other just below the corona to form 5 small slit-like structure. Anthrozoid, this one, anthrozoid swim through this slit, enter into the sheath of nucleus. One of the anthrozoid enter into the egg and fertilization is completed. The zygote create a thick wall, become a oosphere. The zygote falls to the bottom of the pond and germinate after period of rest of few weeks or more than. And next one, the germination of oospore. This is the oospore germinate after period of rest. The nucleus divide by meiotic divisions and to produce 4 haploid nuclei. Oospore divided in 2 unequal cells. Upper cell is the small uninucleate, lower one is large 3 nuclei. These the 3 contain 3 nuclei which degenerate. Later it degenerate. Oospore wall burst open, expose the upper cell and divided obligate longitudinal divisions with produced to the, you can observe, protonymal initial, and another one is the rhizoid. This is the protonymal initial and is the rhizoidal initial. The rhizoid develop from the rhizoidal initial and protonyma initial develop into the erect primary protonyma, which is differentiated into a nodes and internodes later on. And this is the total a new adult plant developed from the new adult plant developed from the primary protonyma. The total life cycle of CHARA is the haplonetic life cycle is present. Okay, did you understood? Today I am ready to teach the very fast, no? Students, are you there?
Madam.
ఓన్లీ ఇద్దరమే ఉన్నాం. యా, అర్థమైంది అనుకుంటాను.
మేడం.
దీనిలో మెయిన్ ఇంపార్టెంట్ క్వశ్చన్స్ ఏంటి అంటే ఫ్యూ ఆర్ ఇంపార్టెంట్. ఎక్కడ గ్లోబ్యూల్, న్యూక్యూల్, ద స్ట్రక్చర్ ఆఫ్ గ్లోబ్యూల్, ద స్ట్రక్చర్ ఆఫ్ న్యూక్యూల్, సెల్ వాల్ ఇంపార్టెంట్. ఓకే,
ఓకే మేడం.
ఓకే, సీ యూ టుమారో.
ఓకే మేడం, థ్యాంక్ యూ.